Assalamu alaikum. My name is Umme Habiba and my ID is 014. I'm from Chemistry Department. The topic of my presentation is disaccharides. Is the contents of my presentation, which include introduction. And in introduction, I'll discuss glycosidic linkage, then important examples of disaccharides like maltose, xylopose, tetrose, sucrose, and trehalose. At last, function of disaccharides. So first of all, we see introduction of disaccharides. Disaccharides are sugars composed of two monosaccharide units that are joined by a carbon-oxygen-carbon linkage known as a glycosidic linkage. Actually, this linkage is formed from the reaction of anomeric carbon of one cyclic monosaccharide with the hydroxyl group of a second monosaccharide. What is anomeric carbon? Anomeric carbon is actually a carbon in that molecule which is derived from the carbonyl group. So in this reaction, you can see glycosidic linkage is formed with the reaction of anomeric carbon of one cyclic monosaccharide with the hydroxyl group of the second monosaccharide. Then important examples of disaccharides, first of all, maltose and sucrose. Disaccharides contain a glycosidic acetyl bond between the anomeric carbon of one sugar and an hydroxyl group at any position on the other sugar. A glycosidic bond between carbon 1 of the first sugar and hydroxyl group at the carbon 4 of the second sugar is particularly common. Such a bond is called a one-fold link. So the glycosidic bond to an anomeric carbon can either be alpha or beta, like this. And maltose, the disaccharide obtained by enzyme catalyzed hydrolysis of starch, consists of two alpha-D-glucopyranose units joined by a 1,4 alpha-glycoside bond. Cellulose, the disaccharide obtained by partial hydrolysis of cellulose, consists of two beta-D-glucopyranose units joined by a 4 glucoside bond. Their structures are given. This is of alpha 1 to 4 glycoside, and this is a beta glycoside. This is maltose and this is cellulose. So maltose and cellulose are both reducing sugars because the anomeric carbons on their right hand glucopyranose units have hemiacetyl groups and are in equilibrium with aldehyde forms. For a similar reason, both maltose and cellulose exhibit mutarotation of alpha and beta anomers of the glucopyranose unit on the right. Like in this, you can see they exhibit mutarotation. Despite the similarities of their structure, cellulose and maltose have dramatically different biological properties. Cellulose can't be digested by humans and can't be fermented by yeast. Maltose, however, is digested without difficulty and is fermented readily. So they have some similarities, and there are also some differences between them. Second example is lactose. Lactose is a disaccharide that occurs naturally in both human and cow's milk. It is widely used in baking and in commercial milk formulas for infants like maltose and soybeans. Lactose is a reducing sugar. It exhibits mutarotation and is a 1,4-beta-linked glycoside. So there are also similarities between lactose, lactose, and maltose. Unlike maltose and soybeans, however, lactose contains two different monosaccharides, D-glucose and D-galactose, joined by a beta-glycosidic bond between carbon 1 of galactose and carbon 4 of the glucose. This is the structure of lactose 1 to 4 beta glycoside. Then example is sucrose. Sucrose, our ordinary table sugar, is probably the most abundant pure organic chemical in the world. Whether from sugar cane 20% sucrose by weight or sugar beets 15% by weight, uh, and whether raw or refined, all table sugar is sucrose. Sucrose is a disaccharide that yields one equivalent of glucose and one equivalent of fructose on hydrolysis. The ratio mixture of glucose and fructose is often referred to as an word sugar because the sign of optical rotation charges are inverse during the hydrolysis of sucrose to a glucose or fructose mixture. It is called inverted sugar. So some insects such as honeybees have enzymes called invertases that catalyze the sucrose hydrolysis. Honey, in fact, is primarily a mixture of glucose and fructose and sucrose. Unlike most other disaccharides, sucrose is not a reducing sugar and doesn't undergo mutation. So it is unlike other disaccharides because they exhibit mutation. This observation implies that sucrose is not a hemiacetal and that glucose and fructose must both be glycosides. This can happen only if the two sugars are joined by a glycoside link between the anomeric carbons of both sugar, carbon number one of glucose and carbon two of the fructose. This is the structure of sucrose. And in this you can see alpha pyranoside and beta fructofuranoside. Then we see some other examples of the trisaccharide like trehalose. Trehalose is also made up of two glucose molecules like maltose, but the molecules are linked differently. It is found in certain plants, fungi, and animals like shrimp and insects. The blood sugar of many insects such as bees, grasshoppers, and butterflies is made up of trehalose. They use it as an efficient storage molecule that provides rapid energy for flight when broken down. So there are many applications of the disaccharides in daily life. We see some their functions of disaccharides. Disaccharides are carbohydrates found in many foods and are often added as sweeteners. Sucrose, for example, is a table sugar and it is the most common disaccharide that humans eat. It is also found in other foods like beetroot. So in this figure, you can see sucrose is also used as white sugar. When disaccharide like sucrose are digested, they are broken down into their small sugars, and they're used for especially for energy. Lactose is found in breast milk and provides nutrition for infants. Maltose is a sweetener and is often found in chocolates and other candies. And Plants store energy in the form of disaccharides like sucrose, and it is also used for the transporting nutrients in the phloem. Since it is an energy storage source, many plants such as sugar cane are high in sucrose. So it proves that disaccharides are also important for plants, but not for only humans. Trehalose is used for transporting some algae and fungi. Plants also store energy in polysaccharides, which are many monosaccharides put together. Starch is the most common polysaccharide used for storage in plants, and it is broken down into maltose. Like there are different functions of starches in the plants. Plant cells can change glucose into starch as a way for them to store sugar with the help of them, and plants tend to store it as starch during the day and sugar at night in the green leaves. So plants also use disaccharides to transport monosaccharides like glucose, fructose, and lactose between cells. Packaging and monosaccharides into disaccharides makes the molecules less likely to break down during transport. So these are some examples of disaccharides and functions of the disaccharides and introduction of the disaccharides. Thank you.